I'm Maya.
I'm Julia.
And this is PDGF signaling. Have you ever been running to the lunch lines for some marshmallow mateys, but then, out of luck, you stumble and you scrape your knee?
It still hurts, and now I have this gross scab.
Well, Julia, that gross scab is keeping you from bleeding alive.
So how does scabbing work?
Well, let's find out.
First, here's a blood vessel. It contains blood cells, platelets, thrombin, and fibrinogen flowing through. Don't worry, we'll explain what each of these is and does as we move through the 3 steps of homeostasis. The first step of homeostasis is the vascular spasm. First, there's a puncture in the blood vessel wall. The blood vessel wall, made of smooth muscle, constricts itself to limit the amount of blood that flows through and escapes. Therefore, less blood leaks out. The second step of homeostasis is the platelet plug, where collagen fibers on the cell wall signal to platelets to stick to the damaged area. Therefore, all the platelets go over here, and we have an accumulation. So, collagen is a type of collective tissue, and platelets produce bone marrow. Platelets release the prothrombin activator, which activates prothrombin to transform into thrombin. Prothrombin is a protein produced in the liver, which is rich in vitamin K. Thrombin is an enzyme in the blood plasma that acts like molecular scissors that cut proteins. It cuts fibrinogen, which is seen flowing through the bloodstream, and it's a glycoprotein complex, and it cuts it into fibrin, which moves on to stage 3. Coagulation is when fibrin fibers, these right here, start to stick and reinforce the platelet clot that was formed in stage 2. They work with the connective tissue to rebuild the damaged blood vessel, and it starts to look like this, and then it's fully healed. This is why we scab.
PDGF is a signal stored in platelet granules. It is predominantly hydrophobic.
PDGF receptor proteins work by derepressing and activating the receptor kinases activity with the purpose of regulating cell growth and division.
The tails of PDGF-A and PDGF-B, the 2 subunits of PDGF, are linked by disulfide bridges and are both rich in positively charged amino acids. They are typically used for distribution/retention of heparin, which is the response when the signal is received. PDGF-C and PDGF-D regulate distribution of latent forms by interacting with other proteins and carbohydrates.
Some advantages and disadvantages of PDGF. The biggest advantage is that scabbing is our body's way to protect us from bleeding out.
A huge disadvantage of the PDGF pathway is that overactivity of PDGF receptor signaling by mutational events is a large contributor to tumor cell growth because of excess cell proliferation. DNA aptamers, or double extracellular parts of the receptor, bind to PDGF isoforms and thus prevent their binding to signaling receptors.
Angiogenesis, discovered by Judah Folkman, is the formation of new blood vessels. This occurs in healthy instances, but also in the formation of malignancies. Tumors attract blood vessels to nourish themselves and sustain their existence. This is why if PDGF pathways go wrong, they can have deadly consequences. However, doctors can treat these cancers with angiogenesis inhibitors, starving the tumor of nutrients and oxygen. There's a diverse list of agonists of PDGF, including imatinib, sunitinib, sorafenib, pazopanib, and nilotinib. Basically, all of the nibs. These agonists inhibit PDGF receptor kinases to treat patients with certain tumors.